Well, thank you to BLOODPAC. Thank you to the Focus Ultrasound Foundation, Susie and Asaf for the invitation. So my name is Nir Lipsman. I'm a neurosurgeon at the University of Toronto at Sunnybrook. And I'm privileged to speak on behalf of a tremendous team here at Sunnybrook, and also fortunate to be a part of multiple collaborations with centers around North America and the world who are looking at focused ultrasound, MR-guided focused ultrasound as a potential tool, not only therapeutically, but diagnostically in the context of brain cancer. I won't recapitulate some of the data and some of the background that you've heard from the outstanding talks so far, but I will just discuss some of our experiences to date and what some of the opportunities are. Just some quick disclosures up front. I own no IP or patents related to anything that I'll discuss today. And I've received grant support from multiple organizations for the work that you'll see. So focused ultrasound itself is a rapidly growing field with expanding clinical and research indications. When you look at the number of publications of focused ultrasound over the last 30 years, you see a gradual or a less than gradual and exponential curve in productivity as indicated by this red line when we looked at the data a couple of years ago. Much of this has been driven by oncologic applications. Now, there are several different applications and mechanisms of action of ultrasound and focused ultrasound in the brain, ranging from thermal ablation, high frequency ultrasound, to opening up the blood-brain barrier with low frequency ultrasound, which is the tool that I'll be discussing today. So it's a rapidly expanding field. And again, oncology has driven a lot of that as a result of the clinical need. So we've heard about the blood-brain barrier and how it represents a challenge. And of course, put simply, it's a challenge not only to get things into the brain, but it's also a challenge to get things out of the brain. So not only are there potential therapeutics that we want to get into in a targeted fashion to treat brain conditions, including brain cancer, most notably brain cancer, but also the acquisition of analytes for pathologic analysis is limited by the BBB. So different techniques have been tried over the last several decades to overcome the blood-brain barrier with variable success, and these range from surgical approaches with chemotherapy impregnated wafers, or other techniques, including molecular mimicry and genetic approaches to try to overcome the blood-brain barrier. There are advantages and disadvantages to each of these, but suffice it to say, none so far have been very effective. So how can ultrasound help? Well, it's been sought as I mentioned, for several decades, a way to transiently and safely overcome the BBB. And the way that ultrasound does this is it uses microbubbles. So these microbubbles, which are a contrast agent that is often used in cardiac ultrasound imaging, is injected intravenously, is circulated throughout the body, ultimately getting to the brain, where they're exposed to focused ultrasound and by absorbing that energy, oscillate about 500,000 times per second. And that leads to a physical disruption in the cells comprising the blood-brain barrier in a transient fashion, such that whatever is co-circulating in the bloodstream can gain access in a highly directed way to brain pathology that is done under MR and real-time image guidance. So the rationale for use in liquid biopsy is that the successful detection of rare events we know is highly dependent on sufficient blood sampling and shedding of biomarkers from targeted tumors. We've seen that the BBB is a major obstacle to do that, whether we're talking about tumor cells or cell-free DNA. So the goal here is, can a less invasive targeted approach to enhance peripheral biomarkers specifically for CNS disease, is that safe and is it clinically effective? So some of the first and earliest data to suggest that you can use focused ultrasound in live preclinical models came from this work, which is now almost 20 years old, 16 years old, showing that focused ultrasound can be used to transiently open the blood-brain barrier in mice models and aid the delivery of Herceptin or trastuzumab, which is 150 kilodaltons. So the typical molecular weight of a substance you can get across the blood-brain barrier is on the order of 400 daltons or so. So 148 kilodaltons and several orders of magnitude larger, showing that, in fact, you can get into the brain safely, and that the MR intensity of gadolinium will actually co-vary with the targeted concentration of the chemotherapy, in this case, Herceptin, that you're delivering. So there seems to be a good link between opening the blood-brain barrier and aiding the delivery of a very large, in this case, monoclonal antibody. And since then, hundreds of preclinical studies have been done in a wide range of animals, from small rodent to non-human primates, showing that in fact, BBB permeability can be achieved, can be done temporarily, can be done repeatedly and safely in a whole host of eloquent and non-eloquent brain regions throughout the brain. And this is work that we reviewed a few years ago, showing that radiographic and clinical effects are quite minimal after this is done. So this is a device that we're using currently, manufactured by Insight Tech. This is the Exablate device. A few features that I'll highlight here. This is the tool that we use to open the blood-brain barrier in human brain cancer patients. The first is that it is a stereotactic procedure in that it is image-guided by real-time image guidance using 3 Tesla MRI in this case. We use cool degassed water as a medium for ultrasound to travel through between the helmet and the patient's head. And we use over 1,000 individual ultrasound transducers, which can be individually steered and directed and therefore specifically target regions of the brain with sub-millimeter accuracy. So this adds up to a stereotactic, and that is anatomically, spatially, and temporally precise means in using real-time image guidance to access brain pathology. So the leveraging some of the preclinical and technical work we showed a few years ago in the earliest experience of FUS BBB opening in human glioblastoma patients. We showed that, and this is an example, in patients with previously resected tumors, we can safely and transiently open the blood-brain barrier, as illustrated by this gadolinium contrast enhancement, which was transient and went away within 24 hours of the procedure at the posterior rim of the previously resected tumor, with no significant clinical or radiographic adverse events. We leveraged that and together with multiple centers around the world, most prominently with Dr. Woodworth, Graham Woodworth in Maryland, a phase 2 trial looking at primary GBM and concomitant delivery of temozolomide in patients undergoing maintenance chemotherapy for GBM. This is an example of the first 9 patients in our study that we treated. And here you can see that one of the objectives of this trial was really large volume BBB opening around the resected tumor. And here you can see in axial, coronal, and sagittal slices that we can open the BBB in a very anatomically precise and quite tailored and personalized fashion to the resection cavity, targeting not only the enhancing rim of the previously resected tumor, but 1 to 2 centimeters beyond that, where, as we've seen, there may very well be tumor cells, likely are tumor cells, and where over 98% of occurrences end up occurring. So this study showed that we can target large volumes without any anatomic restrictions and targeting at least 30 cc's of BBB opening, and the procedure was done awake, non-invasive, as a day procedure with patients being discharged shortly after the procedure in a very well-tolerated operation. We're quite interested as well in targeted therapies for secondary or metastatic brain tumors, in this case, HER2-positive breast mets to brain. So this is a phase 1 trial where we recruited several patients with otherwise progressive intracranial disease with very well-controlled systemic disease who are receiving trastuzumab. And the purpose of this trial was really, again, to demonstrate that BBB opening was safe and reversible, but that we can also target and expand the treatment envelope and target other regions of the brain that are highly eloquent, including the posterior fossa, at the level of the brainstem and medulla, near the ventricle and ependymal surface and near cranial nerve nuclei. This really showed us that, again, no region of the brain is inaccessible to BBB opening, and together with the delivery of chemotherapy, we could do this as a day surgery procedure. One of the novel aspects of that specific trial is that we were able to tag with 111 indium trastuzumab so that we can obtain really the first definitive evidence of the passage of a large antibody across the blood-brain barrier that's been temporarily permeabilized. You'll recall a few slides ago that in 2006, this was shown in an animal model that Herceptin can get into the brain. It required histologic staining to prove that. Here, we can actually show that a tumor, again, at the CP angle that is inoperable, before and after focused ultrasound, here we see a significant increase in ligand attachment to the tumor, indicating, again, the first evidence that a very large molecule, in this case a monoclonal antibody, can get across the blood-brain barrier, bind to the tumor, and in our cohort have a significant effect. So can focused ultrasound be used to enhance analyze for brain cancer? So we've seen now, up until now, the ability of ultrasound to safely open the BBB and potentially get things into the brain, but can we get things out of the brain? So this is work done by Ying Meng, who's a neurosurgeon here at the University of Toronto, who's also joining us today. And what we did as part of our GBM trial that I just showed you is we collected blood work before and after every cycle. And the goal here is, again, can we detect, can we enrich plasma and blood for biomarkers after we've opened the blood-brain barrier? And that's exactly what we saw in this sort of proof of concept pilot trial. Three key findings and takeaway of our work, which was published last year, which is that opening the BBB with focused ultrasound did lead to significantly increased detection of cell-free DNA by at least twofold, and in some much more than that. We also detected a correlation between the extent of BBB opening volumes and the cfDNA that was detected. So there may be a relationship there. The more volume you can detect, that you can open, the more cfDNA you can detect. And finally, that methylation profiling was able to differentiate different patient populations and different samples pre and post opening the blood-brain barrier. We're also conducting a trial of opening the blood-brain barrier and drug delivery in Alzheimer's disease. So we had several samples from that study as well. And again, methylation profile was able to differentiate the different patient populations there, providing very early data, but nevertheless, the first evidence of safe, non-invasive targeted BBB opening in brain cancer that led to enriched signal of circulating brain-derived biomarkers, and that appear to correlate with BBB opening volumes, and that, fortunately bore the signals, the disease-specific signals of a methylation signature for this population. So again, early data that is quite promising, but that suggests that maybe opening the blood-brain barrier can enrich the samples on which we can analyze. What is the next steps for FUS, brain cancer and liquid biopsy? We have a strong interest of applying this across the board in human brain cancer from pediatric to adult populations. We have several open trials in glioblastoma, in secondary brain tumors, and again in DIPG and pediatric brain tumors, and liquid biopsy sampling will feature across the board. So I will just echo some of the messages that we heard earlier about the importance of standardization, the importance of collaboration, and working together with all the centers that are doing this work in order to optimize how we can do these kinds of studies. Why should we do that? Well, because there remain a huge amount of key questions and unanswered questions in the field, not least, which diseases should we be targeting, which stage of treatment should we be looking at, which therapeutic and analyte should we be delivering and detecting, and what are the optimal technical parameters? All of these questions remain to be seen. Important also is the outcomes, and what are meaningful outcomes to patients, and how can we then use outcomes, whether biochemical, clinical, or radiographic, to then ultimately select diseases, to select patients to undergo these treatments in the future. Echoing what Dr. Bagley was saying, a key question in the field is differentiating recurrence from pseudoprogression. And I think, in my view, that is arguably one of the first questions we ought to answer with focused ultrasound and with brain cancer, but there remain many questions. So I'll just conclude with a tremendous gratitude and thanks to the immense team and collaboration that we have here at Sunnybrook at the University of Toronto, Professor Heinonen, whose seminal contributions to focused ultrasound continue, as well as the team that helps us with all of our cancer work and our collaborators around the world. So thanks very much, and happy to answer any questions.